So, what's this talk about? It goes back to September 1998 when I was reading the Sunday Telegraph. To some of you in the audience, I have to explain, I was at my mother's house reading my mother's Sunday Telegraph, and I noticed that on the front page was this big story, scandal of test flaws that led to health scares, saying millions of pounds are being wasted on supposed research breakthroughs because scientists have been using a flawed technique to gauge the significance of their findings, according to a report out tomorrow. This was about medical research, which is my research area, and already a key word, significance, was appearing on the front page, which made me very aware that I had to read on. And turning to the review section of the paper, the front page of the review was the great health hoax. And focusing in and further getting past the garish pictures, the claim is made many scientific breakthroughs are nothing but mirages based on flawed research. They result in wasted taxes, false claims for drugs, and damaging health scares. Robert Matthews, the chief scientific correspondent for the Sunday Telegraph, investigates a global scandal. So just some minor claims being made there about Robert Matthews' findings. So I won't go verbatim into very much that followed, but I will extract what he was writing about. So what is the hoax? The hoax is the frequentist method of statistics. You might have never heard of the word frequentist, but what it refers to is the methodology that 99% of statisticians use 99% of the time to interpret their scientific findings or the findings of their collaborators. So this isn't just a fringe element of statistics that is being accused in this article, it is the main thing. And in particular, he was aiming at the use of p-values. And I will be mentioning, and we will be calculating a p-value pretty soon, so that will be explained to you. But the main thing that I want to emphasize is that he was attacking the whole basis of statistics. And this is a quotation from the article. Combining simplicity and apparent objectivity, he writes, Fisher's p-value method was an immediate hit, and its popularity endures to this day. Open any leading scientific journal and you will see the phrase p less than 0.05 in papers on every conceivable area of research from astronomy to zoology. Indeed, national governments, including ours, still use Fisher's standard to decide whether a new life-saving drug should be approved for use. And this is R.A. Fisher, who was a statistical pioneer. His work was mainly in agricultural research, but he also became involved in medical applications of statistics. And certainly these claims are true. P-values do adorn most scientific papers. Some sophisticated statisticians say that they're overused, but we don't necessarily share in this blanket condemnation that Robert Matthews was engaging in. So I want to illustrate what he was talking about. Now, we're going to do this in two parallel universes. And in one such universe, we are going to think of a clinical trial. And the subject is pain relief. And I'm going to ask 10 volunteers, not from this audience, this is the hypothetical one, who are suffering from chronic pain. And for 1 week, I'm going to give them an experimental pain drug to relieve their pain. And in the other week, I'm going to give them a placebo control. They'll think that they're getting pain relief, but they're not really. To be clever, some of them I'll give the control first and then the active, and some of them I'll give the active and then the control, just to confuse them. And then at the end, I'll say, which did you prefer? Did you prefer, well, they won't actually know which is which, but I will say the green one, because I'm going to make it that the active treatment is in green capsules and the control treatment is in yellow capsules. So, which did you prefer, green or yellow? And if they say prefer green, I'm going to score that as a success, because my active pain relief is more effective than control, than giving nothing. And if they prefer yellow, then I'm going to be disappointed, because they actually prefer the thing that doesn't work. Suppose my new pain relief doesn't work either. What's the probability that my subject is going to say they prefer the green? 50%, a half. And so that's the standard with which I am going to be basing the results on. Now, I think that I have 10 volunteers in this row exactly. And I'm going to ask you to take one block from the bag and hold it up. So that's green. So you preferred the green painkiller. You preferred the green painkiller. I'm very pleased. My drug is very effective. One, please. You preferred the green. This is great. You preferred the green. I'm going to have to show that there are some yellows in here before I finish. There's the green again. Number 6, number 7, number 8, green. We're all green. Number 9. Aha! To prove that there are some yellows, this gentleman preferred the control. It's doing you no good at all, the control. And this gentleman preferred the green. What an extreme result. You can never have probability in your talks because they always do something peculiar. So, we can calculate the p-value now. So, green and yellow blocks. Selecting green is a success. We've sampled 10 blocks. I'm going to use the Greek letter pi to represent the probability of success. And my question is pi equal to a half, or is it greater than a half? Do we think that this drug works? I should hope so, after a result like that. How can you quantify your evidence that it works? Well, the p-value is one way of doing that. So the p-value is defined as the probability of obtaining evidence supporting the alternative hypothesis. And remember, my null is nothing's going on. Null, nothing, nix. The null means nothing is working. The alternative that I'm interested in is that the green is working better than the yellow. The other alternative is also interesting. The green, my new formulation, is actually worse than nothing. It causes pain, but I am not going to entertain as a possibility. So the question is pi equal to 1/2 or greater than 1/2? And the p-value is the probability of obtaining evidence supporting the alternative as strongly or more strongly than that which was observed if there is truly no treatment effect. And here the evidence is summarized by the number of people who preferred green. And the observed statistic is x equals 9. So what's the probability of getting 9 people preferring green, or even 10? Because we've got to take into account we would have been even more impressed if all 10 of you had come up with green as your choice, under the null hypothesis. Well, here is the distribution of x if the probability of success is truly 1/2. There's a 25% chance of getting 5 out of 10 successes. There's just over 20% chance of either 6 or 4. And then the probabilities fall down considerably. Here's the probability of getting 9 if p was really 1/2, and here's the probability of getting 10. So, because I had to prepare these slides in advance, I had to get a slide for every possible value. So there could have been 0 or 1 or 2 or 3. If 4 of you had come up green, I'd have said the probability of getting 4 or more greens by chance is 0.82, and I would record that as my p-value. A large p-value is not convincing. This is a sort of inverse measure. The probability of getting something supporting the alternative hypothesis that the drug works by chance, if that's large, well, it's not very impressive. But it wasn't large because for 9, the probability of getting 9 or 10 successes by chance alone, when the probability really is 1/2, is 0.01074. Now, one of the reasonably controversial aspects about p-values is people who insist on saying, if that's less than 0.05, then we have got a marvelous result. But 0.05 is a useful benchmark. Fisher was asked to suggest a critical value, and obviously he got fed up of people asking him, so he gave them a value, and 0.05 essentially says there's something in that alternative hypothesis. And I would feel that there is something in the suggestion that this drug works, having conducted the experiment. And so the p-value is 0.0107. It's considerably less than 0.05. And now, should we take this drug into bigger trials? Will you invest in my company that is going to market this drug? Perhaps. So that is a good early phase result. So what's the problem? That argument to me is quite logical, and it summarizes the evidence in a way which is quite general, and there are so many different experiments you can conduct and still come up with this p-value. What's the probability of getting evidence in favor of my alternative hypothesis that is as strong or stronger than I have observed. And then all evidence can be measured on this same, by this same measure. Well, the problems that people like Robert Matthews are concerned about is that the p-values ignore plausibility. Would we really think that this drug would work? Depends what went into it. What went into my drug was a bit of coffee, a bit of sugar, and a few tea leaves, and then I put it into a nice capsule. And you might think, well, even given 9 out of 10, I don't think that's going to work to relieve pain. And in more serious studies, and this is one that Richard Peto, who was one of the people behind the discovery of the link between smoking and lung cancer, as he said, a link between smoking and lung cancer is plausible. When you smoke, where does the smoke go? It goes into your lungs. Where do the tumors appear? They appear in your lungs. Seems plausible. A link between smoking and cancer of the big toe, which isn't so much written about but does exist, is still not very plausible. How could that physiologically occur? There are also various problems in terms of doing this calculation of p-values multiple times or repeated times, like after every individual had had their go, I could calculate a p-value. And they're dangerous things when used together. So what's the alternative? The alternative that is best known is the Bayesian method of statistics. And the Bayesian method of statistics is devised to combine opinion with data. In other words, to assess the plausibility of the alternative hypothesis before you begin. And this is gaining popularity in science, very popular in academic statistics departments, but flowing out into everyday use of medical researchers, industrial researchers, environmental researchers. The Reverend Thomas Bayes is a very lucky man. He devised a theorem in probability in the 1700s. And because that theorem is used to calculate the probabilities and the levels of evidence in Bayesian statistics, his name pervades a whole area of science of which he knew nothing. There's only one picture of him, so this always appears. And he discovered a mathematical recipe for working out how to update one's belief in a theory as new evidence emerges. It was a fundamental discovery, for Bayes' theorem gave scientists a way of working out just how much more plausible their theories become as data rolls in. However, before any new finding can be deemed significant, a crucial factor must be included: its plausibility. And now I've got another illustration. And yes, I'm going to require, well, no, I think everybody can see. So I will just I won't have particular volunteers. I will be asking everybody's opinion. But here is some prior opinion. Let me first of all set the scene before I show it to you. So I'm going to show you a jar with pink and black blocks. And my story has changed slightly. This is a new drug. And in my trial, I'm going to give the new drug to everybody. So there's no control in this particular study. Everybody is going to get the new drug. They're suffering from quite a serious disease, and they could either be cured, or, I won't be too dramatic, I'll just say, or not. then we don't become too morbid. And I've got some blocks under that cup, and a pink block represents a cure, and no cure is represented by a black block. Now, you are a consortium of medical experts in this disease, and I'm going to ask your opinion as to the probability that this drug is going to work when given to a patient. And you have what the Bayesians call prior information. And here is the prior information. So look very carefully. That was the prior information. Did you see some black blocks? And did you see some pink blocks? So that's great. What I'm going to be asking you is what the proportion of pink blocks actually was. So think about that whilst we swap to the demonstration. So the first question is the easy question. And I want you first of all to think about it. Don't shout out yet. What was the proportion of pinks in the jar? Okay, so will someone volunteer an answer? What do you think the proportion of pinks was? 70%. About 70%. Somebody else was trying to give me a... Yes? You're going to say 70%. Think of your... think independently, yes? 50%. 40. 60. This really does sound like a convention of experts, doesn't it? 65. I think
it was a quarter. No, it was 67.3. 75.
Okay. So, George, you heard all those opinions. How are we going to put them together? I want one number from this consensus conference. If I say 0.6, that doesn't leave the people who said 0.7 too far out, and it doesn't leave the people who put 0.5 too far out. So I think that this consortium of experts will say 0.6, not 6.0, please, but 0.6. Right. So that's the proportion that you believe is pink, and that's the forecast probability of a cure. And you can see that your opinion depends on your point of view. I like this, literally in this case. And it will depend on the expert's experience. Now, the second question I have to ask is essentially, how sure are you? And we saw that you weren't very sure. And to ask a technical question about how sure are you, I'm saying, what is a probability of cure for which you are 75% certain the drug will beat? Do you get the question? Okay, so 0.5 we are bid there. So this gentleman thinks that there's a 75% chance that the true proportion is above 50%. 0.4. Going down to 0.4. Sorry? 0.65. You can't say that and be consistent with this. We've all got to work on the 0.6. Otherwise, so obviously if it's most likely to be 0.6, it can't be 75% chance that it beats 0.65. So it's got to be a value lower than 0.6. 0.45. Okay, I think we're going to try 0.45. That is obviously the middle of the choices. And I'm going to hope that this works. I think it should. And we're going to click continue. So, yes, this looks sensible. I'm now going to throw back at you some consequences of your choice. First of all, you are 75% certain that the proportion of pinks is greater than 0.45. And because of a model that I'm imposing on this, which I will show you in a moment, I'm also saying that you are 75% sure that the probability or the proportion is less than 0.7. So you're 50% sure that it lies between 0.45 and 0.7. And that has managed to include most of the first responses that we got. A 95% credibility interval for P is still quite wide. You're 95% sure that the answer lies between 0.23 and 0.87. That's a bit of technical difficulty. Detail. The mean value of your predictions is 0.57, and your information is equivalent to having seen 7 patients. And this is an important measure. How certain are you? It's as if you had treated 7 patients and seen a proportion of 60% of them succeed. We're also going to look at the graphics. And the graphics say this is an illustration of your collective belief. Not each individual's, but your collective belief. Here are all the probabilities of pink that it could be between 0 and 1. And this shape is weighting the values that you find credible. So you think values around about 0.6 are pretty well credible. You think that values of 0.9 and above are very unlikely. You think that values of 0.2 or below are very unlikely. Everybody saw some black blocks and some pink blocks. So 0 and 1 are cut out altogether. Shall we proceed with this as your prior? Okay, and I think that that's reasonable, but at this stage, if we were dealing with the real experts, we might have to do this 2 or 3 times. But if we close down that, we can write down the estimate. I'll go for the modal value estimate. So we start with 0.6, and then the credibility interval is 0.45 to 0.70. Now, you may think that this screen is for advertising, but actually it's for a very special purpose. And I'm just preparing the patients for treatment. Okay. And the patients are now all in here. And we've done everything and looked at everything, haven't we? So, update by... Oh, that's no good. Wait a minute. I've got the top of the jar in there. That wouldn't help. So I'll go a bit further back for some clinicians. Would you volunteer to be a clinician? Would you please treat 5 people? So take 5 blocks out of there without looking. Pink, hooray! Black. Black. Pink. And pink. So this doctor has cured 3 of the patients, but not cured the other 2. So the first round of testing has led to 3 successes out of 5. I'm going to put these back. The mathematical experts will know that I'm doing hypergeometric sampling, not binomial sampling, because I'm sampling without replacement, but nonetheless, it's close enough. So, sample size was 5, 3 successes, and we're going to update. And now, as a result of 3 out of 5, well, 3 out of 5 was spot on. Those who believed in 0.6, you can rest assured the data are saying 0.6. The 50% credibility interval has now narrowed, and it's gone to 0.49 and 0.68 at the other end. So that is progress, and we can look at the graph, and that's the current graph, back to the prior. So that's what we believed before we saw the data, and now Bayes' theorem and some modeling using beta distributions, which I'm not going to go into, that's for the extra session afterwards, has led us to change our belief. This is now going all the way up to 3. It's still centering on 60% success rate, but it's narrowing down. There's not much chance, not much belief in low probabilities, and not much belief in very high probabilities anymore. And now I want someone to treat 5 more patients. Two successes. 3 successes in total. 4. Well, this is an excellent doctor. I would be treated by this person every time. That is 5 out of 5. And I'm glad that it's not 3 out of 5 because our estimate is going to change now. 5 out of 5. And so now the modal value, the most likely belief, is 0.73. So you have moved our belief in this drug up to 0.73. And the credibility interval is 0.63 up to 0.78. It's moved up and it's got narrower on what does the graph look like. Is that the bottom one? Yep. Okay. That's the wrong one. Another volunteer doctor. Good. 5, please. 5 patients. So 1 success, 2 failures. And 2 successes out of 5. 2 successes out of 5. So I won't go to him again. 2 out of 5, and that's leading to the modal value is still not gone down to 0.6, it's 0.65, and the credibility interval 0.57 to 0.70. And I usually do 4, so I'll do one more. I'll go to the top man.
Okay. 5.
5, please, yes. 5, 2 out of 5. 2 pinks out of 5, although one had an accident on the way back from treatment. Nearly it was me. So the final reckoning is a modal estimate of, we're back to 0.6. Right, we're back to where we started. And 0.53 up to 0.66. So let's have a look at the graph. Okay. So can you take me through the whole sequence of graphs from prior to final posterior? So this is what we decided we thought of from our prior glimpse of what was going on. And then we had the 3 out of 5, which confirmed the 60% rate, but narrowed the distribution. Then we had the amazing 5 out of 5, and that moved the, yes, thank you, moved the mode up towards 0.73 and narrowed further. Then we had two disappointing cohorts with 40% success rates. We're narrowing all the time, but by the end, we've moved back to being centered on 0.6. And that is how Bayesians learn. Now, I'm not going to do this here, but unlike real scientists, we are able to see what the truth is. And the truth is that there are 35 out of 60 of those blocks, or probability 0.583 is the right answer. So P is 0.583. Sometimes I think I shouldn't do that because in real life you can never look up the answer. But it is interesting, and does this mean that we got the prior right? Who said no? It does not tell us at all about whether we got the prior right, because the prior was what we believed before we started experimenting. And the prior is right if it truly captures the collective opinion of our experts. Our experts might be collectively wrong. That's okay. The data will begin to put them right. And I've done this so many times now. This was one of the best guesses. But we've had some quite amazing runs where the data have taken people a long way from their prior opinions, and people have been worried, my prior was wrong. It isn't wrong if it captures your belief. And then we are, as the title suggests, combining evidence with opinion. And the opinion has to come first and then be modified by the data. So I think I've finished that demonstration, and I want to make, first of all I want to make these points, and then I want to show you what we have been trying to do in practice in real studies. First of all, Bayesian methods versus frequentists. The whole point of this Sunday Telegraph article, and actually, oh, not 2000. Anyway, the whole point of this Sunday Telegraph article was to say that Bayesian methods would exact a higher level of proof and require larger trials than frequentist methods. And that was his whole point about flawed research and conclusions based on a hoax, saying that we're not taking enough account of whether the conclusions are plausible, and we need more evidence before we can reach that conclusion. So Bayesian methods would require larger trials. But other people, for example, a person who works for the Food and Drug Administration in the United States, giving licenses to devices such as pacemakers for heart disease, he wants to use Bayesian methods because they would allow a lower level of proof and because they would permit smaller trials. So Bayesian methods are very good. They are good for people who want larger trials because they can be used to justify that. They're good for people who want smaller trials because they can be used to justify that. I want to briefly talk about the context in which we've been developing methods based on Bayes approaches for early phase clinical trials. This is the phase of experimentation where you're trying to find a dose that has promise, in this case, against cancer. And these are real, I'm going to show you some real data from a phase 1 trial. That is the first time human subjects receive the drug in question. The drug is quercetin, and it's being used in terminal cancer, people suffering from various solid tumors. And this study was conducted by the Cancer Research Campaign in Birmingham. And a whole range of doses were made available, from 60 up to 1,700 milligrams. The patients involved were no longer amenable to standard therapies, and they had various forms of solid tumors. What was of concern here was whether they would suffer toxicity, particularly renal toxicity in the kidneys of World Health Organization grade of greater than or equal to 2. So what happens is you give a patient a course of drug therapy lasting a week or so. If they begin to exhibit toxicity whilst that course of treatment is going on, you have to stop. It hopefully isn't lethal. It hopefully doesn't have long-term effects. But it does mean that the benefit of that course of treatment is probably lost because you have to curtail it. So, toxicity is a bad thing. On the other hand, drugs like quercetin aimed at cancer are intended to kill the tumor. And if they kill the tumor, they're going to kill other things as well, other cells within the body. So some toxicity is to be expected. So if you aim to have 0 chance of toxicity, you probably have got an inactive drug. And the happy medium, or unhappy medium perhaps, is generally accepted to be a dose with a 20% risk of toxicity. So 20% of people will not be able to fulfill their course of therapy, but 80% will. And the dose that achieves 20% toxicity is the dose that we want to use in bigger studies, probably randomized between active and control. And this study was conducted with 18 successive groups of 3 patients. Now, this is a maths talk, and it is the International Year of Statistics, which includes statistics theory, so there is an equation. And some will understand that that equation means that the probability of toxicity is going to rise when plotted against the log of the dose. And remember, the doses were going up 60, 120, 200, 300. They were getting bigger and bigger, and the jumps were getting bigger all the time. So on the log scale, we are thinking this might represent the probability of toxicity, and we want to find the dose corresponding to 20% toxicity. This is how the study was actually conducted. And there are some peculiarities that I don't fully understand. But here's my representation. 3 people given 60, no toxicity. Rise to 120, 3 people, no toxicity. Rise to 200, no toxicity. And we go through 420. What are the doses? We go through 300, 420, 630, and then we eventually get to up to 945. And until we get to 945, nothing bad happens. Then, amongst the 3 people getting 945, someone suffers a toxicity. Traditional approaches say we should repeat 945, but I can only report the real data, and I wasn't there at the time. They went straight up to 1400. 1400 looked all right, so they went up to 1700, and bang, 2 out of the 3 people suffered toxicity. So they came down the other side, finding more toxicities at 1400. Eventually, 945 was what they zeroed in on. They did visit 630 again, but 945 was what they chose. Now, a Bayesian approach to dose finding goes like this. Find a prior, and that prior has to be about where that S-shaped curve goes up and how steeply it goes up. And we have to ask experts like yourself for their opinion about those parameters. We also make sure that the prior says the best dose to give the first time is the lowest dose, because we know that that's where experts always start. First, 3 people receive dose D1. We then look at what they did, and we then do an analysis using Bayesian methods coming up with a posterior in a way similar to what we have just been doing now after groups of 5 patients. And then we choose the doses for cohort 2. Cohort 2 receives the chosen doses, and so it goes. And when we apply a particular form of that, this is simulated. It says start at 60, and we take the first 3 real people who had 60 and find that they didn't have a toxicity. It then skips over 120 straight to 200, and those people are all clear. It then skips up to 420, they are clear, up to 630, and then up to 945, where that toxicity is waiting because we know that this is the real data but rearranged. But our version doesn't plow on up to 1400 after that signal of toxicity. It says, hang on a minute, let's go down and try 630 again. And we know that the real data said number 5 on 630 had a toxicity, so it gets rather stalled. The circles, if you can make them out, are there because we had to make up some data. There were no more than 5 real people on 630. And we made up the data assuming that they didn't get a toxicity. Then we go up to 945. We do visit 1400 and 1700, but the end, the conclusion, is the same as the real study with 945, but in a way that I believe has been rather safer, more sensitive to toxicities, and learning as it has gone along. So my conclusions for the talk is that p-values have their flaws. Robert Matthews is right to point out that this methodology isn't perfect. But the claims that he is making are about flaws that those using the methods have known about for many years, probably ever since R.A. Fisher himself. All of the tools that statisticians can use have their problems, have their limitations. And so do the Bayesian methods. And one is the difficulty in choosing a prior. We did quite well, I think, today on that simple exercise. But when there are multiple parameters and they're all related to one another, it becomes more difficult to reliably choose a prior from a consensus meeting like this. So I think statisticians need every tool in the book, and therefore we should use frequentist methods when we can. We should use Bayesian methods when they will add something. Both are part of the big picture and neither give the whole thing. And scientists should evaluate both the frequentist and the Bayesian properties of their procedures, and therefore they shouldn't use a method just to make it harder to claim that a treatment works, nor, conversely, to make it easier, but to have procedures that have an appropriate error rate and an appropriate way of reflecting information. So, thank you for your attention. I want to acknowledge that the filming of these sessions have been made possible by a donation to the university from Joseph Devlin, who was a graduate with a BA in economics in 1981, and he contacted the university earlier this year suggesting a donation, and we suggested this use of it so that these talks and probably the back of my head will be immortalized on our website for some time to come.